Here at the Dana-Farber Cancer Institute, investigators are leading over 700 clinical trials across a variety of settings, including over 60 focused on breast cancer. One such study is comparing the antibody-drug conjugate trastuzumab emtansine, or TDM1, with paclitaxel and trastuzumab. Principal investigator Dr. Sarah Tolaney met with us to discuss this study.
ATEMPT trial is a trial that's a randomized trial comparing trastuzumab DM1 to paclitaxel and trastuzumab. It's for patients who have stage 1 HER2-positive breast cancer. And for those patients, they're randomized in a 3-to-1 fashion, either to the TDM1 or to the paclitaxel and trastuzumab. So they receive, if they get randomized to the TDM1 arm, they get TDM1 every 3 weeks for a year. And then on the paclitaxel and trastuzumab arm, they get 12 weeks of paclitaxel and trastuzumab, followed by 9 months of trastuzumab monotherapy.
The ATEMPT trial hopes to build on the recent APT study, which examined adjuvant paclitaxel plus trastuzumab. This study demonstrated promising results. However, researchers believe that TDM1 alone could be superior. For further insight, we interviewed one of the APT study authors, Dr. Harold Burstein, about the study that set the groundwork for the ATEMPT trial.
We know that women who have small HER2-positive breast cancers have a generally good prognosis, but because of HER2 overexpression, they can be somewhat riskier than you might imagine, even though the tumor is very small. And so as a way of coming up with a suitable regimen for these women, our group led a study we called the APT study, the adjuvant paclitaxel trastuzumab study, where we took about 400 women at our center and many centers around the country who participated in this trial with us and treated them with 12 weeks of paclitaxel plus trastuzumab, followed by a year of maintenance trastuzumab. These patients all had very small HER2-positive breast cancers. But what was important was we showed that there was an outstanding result. There was really only a 2 to 3% risk of cancer recurrence through 4 or 5 years of follow-up, which we thought was very good, and this is a reasonably well-tolerated regimen.
The ATEMPT trial hopes to show that TDM1 is less toxic than the paclitaxel-trastuzumab combination, while demonstrating an advantage in terms of disease-free survival.
There are 2 co-primary endpoints. One of them is to compare the toxicity between the 2 arms, and with our hypothesis being that the TDM1 arm may be better tolerated than the paclitaxel trastuzumab arm. And another endpoint is to look at the disease-free survival within the TDM1 arm itself. So we had done a prior trial called the APT trial that had looked at the paclitaxel trastuzumab combination in a very similar patient population, and found that patients did extremely well with that regimen. The 3-year disease-free survival was 98.7%. So we felt that we may want to try to find an agent that may be even better tolerated for this low-risk population, and we thought TDM1 seemed like an attractive drug, which in the metastatic setting there's a trial suggesting it may be slightly more efficacious than paclitaxel and trastuzumab. So that's why we designed this trial, which is sort of a follow-up study to the APT trial, really to compare the toxicity between the 2 drugs.
What's exciting about TDM1 is it's the same antibody as trastuzumab that targets the HER2 protein, but they have linked to the antibody the actual chemotherapy moiety. And so it's like you use the antibody to sort of deliver the chemotherapy like a smart bomb right to the tumor. And the beautiful thing about it from the patient's point of view is it does not have many of the typical chemotherapy side effects. It doesn't make your hair fall out, it doesn't make your blood counts get low, it doesn't give you neuropathy or tingling in the hands and the feet. So it's a very well tolerated product. And so the study is designed to show that these results will be as good with TDM1 as they were with paclitaxel trastuzumab, and hopefully show that it's an even better tolerated regimen.
This study is currently enrolling patients at the Dana-Farber Cancer Institute with an estimated enrollment of 500 patients.
Patients need to have stage 1 HER2-positive disease, so they need to have tumors that are under 2 centimeters. They are allowed to have 1 micrometastasis in 1 lymph node, and they have to have good cardiac function, so they need to have an ejection fraction over 50%. And we do require central confirmation of the HER2 status, so all tumor gets sent to a central lab for HER2 testing, and sometimes that does cause a slight delay because it takes 7 to 10 days for a turnaround before patients can enroll.
Patients with excessive alcohol intake, defined as 3 or more beverages per day, will be excluded from the study based on early reports of potential liver toxicity.
There has been some reports of TDM1 liver toxicity, so we want to make sure that patients who are on study aren't patients who are drinking lots of alcohol. This was actually something that was mandated by the FDA for this particular trial.